This is episode 171 of the Stem Cell Podcast, The Heart and Human Development with Dr. Benoit Bruno. Hey everybody, this is Dr. Daylon James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. We were surrounded by them. Like many stem cell researchers, Arun and I attended the 2020 ISSCR virtual meeting, not really surrounded by them. We were virtually surrounded by them, which is totally meaningless. We were staring at a screen. But we were there virtually this past week, and we have some great interviews in store for you with some of the meeting's top researchers. That's including Doctors Madeline Lancaster, Christy Red Horse, and Martin Pera. If you missed out on the meeting, you can also check out our daily ISSCR video roundups, where we summarized some of the most interesting research that was presented at the meeting. To watch those, you should visit stemcellpodcast.com/ISSCR2020. Check it out. That's us in a video format. Please forgive my hideous visage. But Arun is a pleasure to look at. So
thank you.
Get over there. Today, we have Dr. Benoit Bruno from Gladstone Institutes on the podcast to talk about his research into the mechanisms of heart development. We've also got our usual roundup of recent highlights and stem cell news. That's coming up.
But first, we have to remind you guys about Muscle Cell News, one of Stem Cell's free weekly scientific newsletters, something I actually take a look at every day. Really good heart stuff in there too. Muscle Cell News summarizes all the latest research, news, jobs, and events in muscle cell research and delivers it right to your inbox every Monday. So save time and keep current with Muscle Cell News. And you can subscribe for free at www.musclecellnews.com. So we're going to get into the roundup here. And the first paper I'm going to talk about is coming from the lab of Carl Gregory. First author is Ian McNeil. Title of this paper is Characterization of a Pluripotent Stem Cell Derived Matrix with Powerful Osteoregenerative Capabilities. And this is published in Nature Communications. Here we're talking about bone and bone repair. And we know that most broken bones can be fixed with like a cast, for example, right? But more complicated fractures actually require treatments like bone grafting. So this is coming from researchers at Texas A&M who have created superior bone grafts using iPSC-derived cells. They found that these grafts can create really good scaffolds that are needed for the bone to actually regenerate at the site of repair. So the general premise here is, instead of using mesenchymal stem cells, which can be somewhat heterogeneous in terms of their differentiation, in terms of their derivation, they're using iPS-derived human mesenchymal cells. And the thought here is, one, you can scale them up very quickly, and two, they're able to actually create a matrix, an ECM, really harness the extracellular matrix from the iPS-derived mesenchymal stem cells to repair bone. And so how do they do it? First, they confirm that their iPS-derived human mesenchymal stem cells are sharing the characteristics of their primary mesenchymal stem cells. Next, they demonstrated that the iPS-derived mesenchymal stem cells, the osteogenesis in these cells is regulated by Wnt signaling and also PPAR gamma signaling. And when you actually inhibit PPAR gamma, that's increasing the osteogenesis of these cells. The next thing they did was their kind of the crux of the paper, which was use extracellular matrix derived from purified monolayers of these iPS-derived mesenchymal stem cells, and identified that they have really enriched levels of collagen, as you might expect. It's ECM, right? And if you graft this ECM onto a mouse model, you can really substantially improve bone repair. If you get rid of the collagen in the ECM, you would expect, it results in osteogenic deficiencies. So the bone isn't able to grow back quite as well. So it's a nice study that's spanning the gamut from basic science all the way to an in vivo model, from in vitro all the way to in vivo, then makes sense, right? Bone is something that this is a very widespread phenomenon, bone repair, right? And a lot of people have fractures. I've had broken bones back in the day, and anytime you can figure out a way to improve bone healing, I think this really has a ton of translational potential too.
I've never broken a bone. Isn't that amazing? So,
Really? Wow.
I don't need this stuff. I'm just not interested. But yeah, I'd probably just jinx myself. The thing I think is interesting about this is a couple things. One is that this idea of using a cell source to generate a kind of acellular substrate. So there's kind of I think a broader potential for this. Maybe if you take the cell component out of it, but you have a material, even an IPS derived material that can then generate unlimited matrix for you, that could be really a robust system. But in terms of like the mesenchymal stem cell differentiation, I feel like, the whole idea of a mesenchymal stem cell, while at first there was a kind of negative stigma, or at first everyone was crazy about it, and then the stigma grew. Now I think we're rethinking what even defines a mesenchymal stem cell. It's kind of a catch-all definition. We've talked about that with some of the guests. So I'd be interested. And I think what's good about this is like you get primary MSCs from a patient, depending on the tissue. They could be, even you get it from the same tissue, a different day, different prep. Who knows? There could be differences. Whereas a iPSC starting material, I think the benefit there is maybe you could get a more reliable derivative, but still, and I know they do go some ways, as you said, in characterizing how much like the true bona fide MSCs, their IPS-derived MSCs are. But I still would ask the question, like, what MSC do they correlate to in the body? What is the degree of difference between some cells within that prep or from passage to passage? Because that might affect, of course, the constitution of that ECM. So a few questions I have there.
Yeah, it's valid questions, all of them. When it comes to the specific subtype of MSC, I don't know if that's actually well characterized here, but it's definitely a valid point. Really the differentiation of MSCs from iPSCs is somewhat heterogeneous, and you probably have a mixed bag, just like you have a mixed bag of cardiomyocytes when you differentiate them. But one thing actually you referred to here that I thought was really relevant, was the possibility of using these cells as a factory, right? You're not necessarily using the cell itself as your therapy and your regenerative approach here, but you're using the ECM that's derived from the cell. So in essence, what you're trying to do here is use these cells as a factory for producing ECM, not unlike what we talked about previously a few weeks ago when it came to using the cerebral spinal fluid from those organoids. Potentially, if you can scale that up, right? What if you have like an unlimited supply of cerebral spinal fluid if you just pop the little bubbles in those organoids, right? Maybe you can use those organoids as a factory for CSF, just like you can use these cells as a factory for ECM. Who knows?
Yes. There's a lot of potential. I like this idea of cells as factories because it's much more, I think, low-hanging fruit clinically and also from an investigation standpoint. So I'm with you on that. You talk about, the identity, these MSCs. There's another age-old debate vis-a-vis identity in tumor cells and tumor plasticity. It's been well documented, and specifically as it relates to this vascular mimicry. It's an idea that was put out years ago. I mean, when I studied endothelial cells from pluripotent stem cells, it was a big factor there is how much like actual bona fide endothelial cells they are, whether they can recapitulate this vascular mimicry that we see in tumor cells. Because there is clearly plasticity amongst tumor cells because they do whatever they got to do to adapt to the environment. And this has been well described in terms of like epithelial mesenchymal transition, or EMT, which is critical in tumor growth and metastasis in all epithelial-derived carcinomas. But there's also this vascular mimicry or tumor endothelial transition that's been reported in glioblastoma and other cancers in vitro and in vivo. And this is interesting because the tumor cells, they shed their tumor-related profile or phenotype, and they adopt this endothelial phenotype. And the relevance to this is high because the vascular channel, that's a means by which you get metastasis to distant sites. So it stands to reason that if it's these endothelial cells and vascular mimicry that's mediating it, you could target that process and mitigate the degree of metastasis of many cancer types. So Lydia Koss, who's at Florida International University, no coincidence in Miami, she's studying melanoma, a lot of sun over there, so she's probably getting ahead of it. They use a spontaneous mouse model of melanoma and show that there is indeed this phenotypic plasticity. They do a lot of lineage tracing and elegantly and rigorously show that there's this phenotypic plasticity whereby the melanoma cells, they colonize this intravascular niche and become indistinguishable from surrounding endothelial cells. And they just hang out there. They, are quiescent and chilling out until they can then be mobilized then by an endo-MT or endothelial to mesenchymal transition, where they then extravasate into the tissue again and cause these metastases. And then they go on to show that this is not only evident in these spontaneous mouse models, but also in human melanoma samples and xenografts. So they really extend this to the clinical relevance by showing that this isn't just a phenomenon in an experimental setting, but that also human tumor cells seem to be doing this very same thing. So a real, I think, important result that probably going to have some implications, not just for melanoma, but for a lot of cancers, including glio, where you've noted this kind of vascular mimicry phenomenon.
Yeah, this is a story about transdifferentiation, and we've been talking about that quite a bit on the podcast, the possibility of cells transforming from one cell type to another. I don't know about you, but this to me, this is a little scary. This is a little scary because there's the possibility of cells just potentially just hanging around and then ultimately transforming into a cancerous cell down the road. And you actually before we actually recorded this, you were telling a story about a family member of yours who had recovered from cancer and then unfortunately had a car accident and that cancer returned. So perhaps there is a really strong environmental element here too.
Yeah, I think there's a lot of anecdotal evidence of injury or some other kind of inflammatory disease conditioning causing relapse of cancer. And that's what's scary, as you said, about this vascular mimicry phenomena is the latency. I'm starting to think of cancer like not of all the cancers, that you get and treat, but all the cancers you don't get. You die with all these latent cancers that are just hiding out. And maybe the means to avoiding cancer and living a long life is just not reactivating all those latent little mimics in your vasculature.
Yeah, it's tricky because there's such a strong environmental component, whether it's, the things that you eat. And we know that, of course, that's a huge area of study here these days when it comes to the microbiome and the effect that actually it has on reactivating or activating cancer potentially. So definitely a lot of work that needs to be done here. Next paper we're going to talk about is moving on to the eye. So we're talking about modeling glaucoma in iPS-derived cells. This it's glaucoma is a condition that damages the eye's optic nerve, right? And it's something that's pretty prevalent. It's something that's becoming more prevalent among the aged portion of the population, right? The aging population. And it's usually the result of high pressure inside of the eye. And over time, the pressure can actually erode these retinal ganglion cells, which might lead to vision loss or potentially blindness, right? If I had to pick one sense that I definitely would not want to lose, it's my sight, right? So definitely very important work here. And there's a bunch of animal models, which of course have been developed to understand glaucoma, but folks here at Indiana University are reporting using human pluripotent stem cell models where they can actually analyze the deficits in cells that are damaged by glaucoma. So it's an iPS-derived glaucoma model, iPS-derived retinal ganglion cells. Pretty straightforward disease model here. It's in stem cell reports. First, they generated some CRISPR-modified iPS lines for the OPTN gene, which is a gene that's critical for eye development and also for the onset of glaucoma. They did an E50K mutation here. They also had some isogenic controls. They saw that this mutation does not affect the early stages of retinal differentiation, but it's later on. It's at the later stages of the maturation in these iPS derived retinal ganglion cells where they actually have neurite defects. And they did some pretty cool neurite outgrowth tracing here. Really, really neat figure in figure 3 that you can check it out. The next thing they did was characterize these mutated cells and this E50K mutation, and they saw that these retinal ganglion cells actually had a downregulation of various RGC-associated transcription factors, general RNA sequencing here. But I think the exciting thing here was the functional analysis. Anytime you're trying to do a disease model using iPS-derived cells in vitro, whether it's cardiomyocytes, neurons, whatnot, I think showing a functional difference between the control and the cells that you're interested in is, I think, the really powerful thing. And here they're actually showing that these OPTN mutated cells, these mutated retinal ganglion cells, actually have an enhanced excitability. Okay, so that is their functional output here. And finally, wrapping things up with some more RNA-seq, they're able to show that there's differential gene expression between the control and the OPTN cells. So this is a relatively straightforward disease model teaching us something about glaucoma, which is something that a lot of us experience as we age. And as I mentioned, anything that you can do to better understand sight and how we lose our sight is needed, because it's a scary thing, losing your vision, and we need to better understand how that happens.
What jumped out at me when I read the title of this, retinal ganglion cells with the glaucoma OPTN mutation exhibit neurodegenerative phenotypes when derived from three-dimensional retinal organoids. So I kind of just keyed into the title and thought that maybe that meant that when they're from 3D organoids versus if they're derived from monolayer. Do you have any idea if that's true? Is like the organoid 3D differentiation? We always talk about that, how 3D allows you to recapitulate not only like cell types and molecular environments, but also like functional elements that aren't present in monolayer. Is that essential in this context? Do you have to have 3D? Do you know?
Yeah, I don't think they did that direct comparison between two different differentiation protocols. As far as I understand, these days, a lot of folks have shifted towards 3D differentiation for optic differentiation, like retinal ganglion cells. It's just perhaps a more efficient differentiation process. But certainly, the ideal experiment would be to derive these mutated cells not only in the 3D context but in a 2D context and see if you can see that same disease phenotype, right?
Yeah, you should do that experiment, Arun, and let me know how that works. Okay.
Last time I checked, I'm not an eye guy, but I'll get on that.
Hey, man, you can always grow. So yeah, this is going totally into the left field here because it's not really, strictly speaking, a stem cell story, but it's a blood story that I love. Also, it's a bit of a callback. Also, our guest coming up in a few minutes, Benoit Bruneau, was at the ISSCR, and I saw this speaker, also Michelle Satterley and Scott Lowe. I saw the story. I saw Michelle talk about this story in part. It was in Nature a couple weeks ago, so I'm covering it. Okay, listen up. This is also a story that you kind of, led into, and I'll come back to that. It's about senolytic agents. Okay, a lot of people are excited about these senolytic agents that selectively eliminate senescent cells, and this kind of started almost a decade ago with work out of the Mayo Clinic that showed you could use this ink attack, which would selectively eliminate P16 INK4A positive cells, which that's a hallmark of senescent cells. And you would get apoptosis of those cells, and it led to longevity in these mice. So you clear the senescent cells, the mice do well, presumably the humans do well. So then, just like a few months ago, I think you did the roundup story from Jonathan Epstein, where they used CAR T cells. So the idea there is let's take CAR T cells and we'll target them against senescent cells. In this case, they were targeting against senescent cells in the heart, fibrotic cells to target cardiac fibrosis. And they targeted this fibrillary acid protein or something, FAP, and they showed it was good for the heart. Okay, no coincidence. This paper came out, that Jonathan Epstein paper came out like September 18th. Michelle and Scott said, we're scooped. That's when they submitted
Hmm.
this paper to Nature. All these papers, by the way, were Nature. The senescence was Nature with the Incotech, Nature with Epstein. Now, this is another Nature paper out of Memorial Sloan Kettering. Like I said, Michelle Satterley and Scott Lowe are the lead. And what they show, it's basic. It's just like what you did. They first looked for proteins that were uniquely present in senescent cells. And this is key because they use these three different models that they said robust models of senescence, which was this mouse lung adenocarcinoma that's triggered by therapy to senesce. And they used a oncogene-induced senescence in mouse hepatocytes and culture-induced senescence in mouse hepatic stellate cells. All right, so that's lung and 2 liver, right? And by doing this in comparison to normal cells, they found that there was this PLAUR, okay? So urokinase type plasminogen activator receptor that was unique in senescent cells. They also showed that it was unique to human senescent cells. They generated some CAR Ts to that, and lo and behold, it worked. These CAR T cells, they could attack the Plower expressing cells in vitro, in vivo, and here's was the functional output here that was so exciting me. A Nature paper was that they extended the survival of mice with lung adenocarcinoma, okay, lung, and they restored tissue homeostasis in mice that had liver fibrosis that was either induced chemically or by diet. Now, I'm not trying to take away because like that's huge. Lung and liver, those are a big deal. So if they worked for just that, it'd be enough. But I think, it's no coincidence that they targeted Plower, this urokinase type plasminogen activating receptor, based on these 3 models that were lung, liver, liver. And then they tested it in the lung and the liver and showed that it worked. So while this is a powerful idea for a very directed therapy, I think you still have to wonder about whether or not there's off-target effects, number one, or whether or not this approach specifically with this urokinase type plasminogen activator receptor is going to be suited to kind of senescent cells throughout the body, or if it's really just going to work in the liver and the lung in these specific contexts. But still very exciting work. The crowd went wild at ISSCR when they presented this. It was part of like a spate of these new CAR T based like cell engineering, super crazy therapies that are going to be they're like the standard in the future, I'm guessing.
Yeah, this is immunotherapy taken to the next level. And you talked about the other paper that we discussed on the podcast not too long ago, using CAR T to target fibrosis in the heart, and this is kind of an extension of that, right? We're looking at senescence, cell senescence specifically here. I mean, the thing that really excites me about this is because there are so many other disease phenotypes and just conditions that are associated with cell senescence, not only in these examples, but also Alzheimer's, for potentially progeria and aging have various associations with senescent cells. And if you can co-opt these CAR T approaches to actually target these other senescent populations, then I think that's widely applicable across the board.
Yes, that's the idea, is eternal youth. But, one thing to consider is that there is no precedent for that in nature. We're kind of outside of natural physiology here. And it's important to note that senescence does exist for a reason. There are some compartments where senescence has an important role molecularly, functionally. So we have to watch out about clearing those. But as you said, I mean, this is I think the implications for this are broad, and it's just a matter of finding an alternate cell surface localized factor that's unique to whatever niche you care about. You can target anything. This CAR T, we started talking about it was just blood-borne cancers, and now it's everything. So it's really, really exciting, and I'm not going to stop talking about it, Arun. So brace yourself. But we'll take a brief respite into the heart because that's your thing. I like the heart too. Coming up in a few minutes with Dr. Bruno. But first, I have a brief message from Stem Cell. Do you work with human pluripotent stem cell-derived cardiomyocytes? Our guest today does. Arun does. I do. You should use Stem Cell's StemDiff cardiomyocyte media and supplements to differentiate, enrich, expand, and cryopreserve functional HPSC-derived cardiomyocytes. StemDiff cardiomyocyte media is compatible with human embryonic stem and induced pluripotent stem cells. And the resulting cardiomyocytes can be used for disease modeling, drug discovery, and cardiotoxicity screening. Learn more at www.stemcell.com. /StemDiff-cardio. All right, guys, today on the podcast, we have a special guest, Benoit Bruno, who is director of the Gladstone Institute of Cardiovascular Disease. The Bruno Lab investigates the transcription factor networks that regulate lineage, commitment, and differentiation during heart development and model congenital heart disease using mouse models and human-induced pluripotent stem cells. Dr. Bruno, thank you very much for joining us today.
It's my pleasure. Thanks for having me.
The pleasure is ours. Let's start with the kind of broad scope here. When you think about stem cells in the media, I think, the lay public and media like to focus on the regenerative aspect of stem cells for good reason. It's a huge boon for translational regenerative medicine. But that's mostly for the purpose of treating adults. And congenital heart defects, by contrast, the most common of all birth defects. And we've come a long way surgically in treating newborns, even fetuses in some cases, rarely. But why don't you start off by giving us an overview of how you leverage pluripotent stem cells? I mean, we talk a lot about how you can use it for a heart patch, or heart organoids, or modeling disease and pharma, oncology, and all that stuff. Why don't you talk about how you can leverage pluripotent stem cells toward understanding and treating heart defects?
Well, so that's a really tricky one. And it's something that we struggled with for a long time, because congenital heart defects are a three-dimensional structural defect in the formation of an organ. So how do you model that using in our case, iPS cells? And the question we've struggled with is, can you even glean anything from iPS cells to understand congenital heart disease? Because congenital heart disease, and we understand the genetics of it, it largely arises from mutations in transcriptional regulators, so transcription factors, histone modifying enzymes, so genes that, proteins that regulate gene regulatory networks broadly. And so you can imagine a number of scenarios where in a fetal heart that has a mutation in a transcription factor, that either the activity of the impaired activity of that mutated transcription factor affects the formation of just a small subset of cells, let's say the atrial septum or the atrioventricular node or the ventricular septum, and that there's only those particular cells that have a disrupted gene regulatory network. If we were trying to model that using iPS cells, 2D in a dish with directed differentiation, we don't even know if those particular cells exist in an iPS cell-based differentiation system, or could we even find them if they were there, but there were only 5% of the population. So is it even worth the trouble to try to model congenital heart disease in a dish? The other possibility is that these transcription factor mutations cause a broad dysregulation of gene regulatory networks in all of the cells in which they're expressed, but that disrupted gene regulatory network only manifests itself as a structural defect in a particularly sensitive part of the heart. So in that case, the iPS cells would be perfect. So we have a preprint that's now in bioRxiv, where we've tried to model the loss of function of one of our favorite transcription factors, TBX5. Those are the first mutation associated with human congenital heart disease. We've been working on this for a long time. And so we modeled its haploinsufficiency in iPS cell models, and we're delighted to find that both of the potential models that I explained are actually right. And we're able to achieve that using a single-cell RNA-seq, where we find that there's broad dysregulation of gene regulatory networks in cardiac myocytes that are derived from the mutated iPS cells. But in many cases, we only find gene disruption in subsets of the cells in the dish. So it's imperfect, but it has been able to get us to an understanding of human congenital heart disease, which we were never able to get to until now. Now what we want to do, of course, is then say, okay, what we've learned in iPS cell models related to congenital heart disease, can we go back to a 3D organ? Can we go back to the mouse and see whether those things play out in our mouse models? And we're in the process of doing that.
So Benoit, we're of course fans of iPS cardiomyocytes here on the show. We recently had your colleague Deepak Srivastava on the show, and I guess all three of us are basic cardiovascular biologists who are using iPS cardiomyocytes. One thing we didn't really discuss with Deepak though is epigenomics, and this has become a really big focus in your lab, where you guys look at chromatin remodeling complexes and the role of CTCF to regulate 3D chromatin structure, all in the context of studying cardiac development. And the traditional view is that it's really the cardiac transcription factors like TBX5 that you mentioned, the GATA family, that are doing the real heavy lifting during cardiogenesis. But of course, you have to view that in the context of the epigenome and chromatin dynamics too. So talk a little bit about the epigenome in the context of heart development and how misregulation might contribute to congenital heart disease. So what have you guys learned about that?
Yeah, that's really fascinating. I mean, I just like Deepak, I grew up with DNA binding transcription factors, and I still think that those are sort of the most powerful instructors, right? They guide the way. They land in a particular place, and they tell the chromatin remodeling machinery and histone modifying enzymes, come over here and do your thing. But with that said, the transcription factors are also pretty inert molecules. They sit there, and then their job is to then bring in the rest. So what is the rest? And how does the rest play in to gene regulation and congenital heart disease? The thing that I find is really interesting about all of these mutations in congenital heart disease is that they're all heterozygous mutations, right? It's the dominant mode of inheritance, meaning that the dosage is really, really important. And so you could understand how the dosage of a transcription factor might affect gene regulation, right? 50% loss of occupancy, or it goes only to the right place 50% of the time. But what about the histone modifying enzymes? What about the chromatin remodelers? We generally think of those as being, they're just there. They go everywhere. They touch everything. They're not considered to be very specific. Now, we've found in our studies of the SWI/SNF-like BAF complex chromatin remodelers that in fact the function of these chromatin remodelers is exquisitely site-specific. We don't understand exactly how that happens. We think it has to do with the subunit composition of the complex and how that's dynamically regulated from cell to cell, and that somehow, and we don't understand it yet, that dictates the preferential recruitment of those complexes to one locus and the other. Now, you mentioned the epigenetics, and that becomes perhaps even more nebulous because the epigenetics are measured quantitatively. We talk about increased or reduced ChIP-seq signal for a particular histone modification, but how does that actually translate to altered gene expression? I don't think that's really, really understood. And the final point is that you bring up CTCF and 3D chromatin interactions. I think that when we're thinking about the histone landscape and, for example, enhancer promoter interactions, that I think there's emerging evidence that's where the connections are being made, that the histone modifications and how they create a potential for the proximity or the interaction, or however the phase transition, if you want to use a sort of the super enhancer of the day type phrase, those preferential interactions, I think, are quantitatively regulated. And I think that's how the epigenetic landscape modulates specific loci in a quantitative way.
We're talking super, super deep, basic mechanism, molecular bits and pieces here. And I mean, that's what they get at the Gladstone. It's a powerhouse, both basic and translational as well, of course, cardiovascular research. We've had members in regular rotation, as Arun mentioned there. We had Deepak Srivastava, also Todd McDevitt joined us. What draws such, the brightest minds, what draws you all to the Gladstone, or what's the common denominator you think there? Is it intellectual freedom, as they talk about? That pie in the sky in science, is it the ultimate freedom, or is it just because you have such a brain trust? Talk about that.
I think there's three major aspects. I think one is definitely the intellectual freedom, and specifically the notion that we're basic scientists. And we talked, I was, Arun and I kind of nerded out on transcription factors for a second and epigenomics. But really, what we're focused as overall at the Institute, and whatever disease it is that we're focused on, whether it be heart disease or neurological disease, we're focused on basic science towards understanding and curing disease, right? So it's not like, let's say, a biochemistry department where we want to understand molecules and phosphorylation of this particular pathway or that. We want to understand that, but we want to understand it really with the end game being we want to understand disease and we want to be able to generate potential therapies for those particular diseases. So that's number one. I think we all come together with a notion that we want to understand disease from a basic science point of view, and we want to make a difference for people afflicted with those diseases. The other is culture, right? I mean, we choose people who join us really, really carefully, and we have a culture of collegiality, of mutual respect. We're in a place where we celebrate one another's success. We go out of our way to help our neighbors succeed, and it takes a special place to empower that, to not feel that we need to be going after the next big thing. We're here to do the best science we can together. And it takes a really special place to do that. And the other thing, which is really just a practical thing, is that we're a medium-sized independent research institute, and that gives us a way of doing things that are nimble and fast, and we can do things the way that we want to do them. And if we need to change tech, we can just decide to do that. We can do things the way that we want and not be encumbered by a gigantic administration with cumbersome rules and so on. So it's a combination of being small and nimble and adaptable, and really excited about pushing the envelope at all times.
So the Gladstone is certainly a powerhouse for cardiovascular science. And as you just mentioned, you're not afraid of thinking outside the box. So we got to talk a little bit about a pretty wild project that your lab did recently, which got a ton of press, one of my favorite science projects of all time. So in a collaborative effort, your lab sequenced the genome of the Komodo dragon, showing that it has positive selection in gene pathways related to energy metabolism and cardiovascular homeostasis. And then you showed that this selective pressure might have influenced the blood genes that actually help Komodo dragons evade the anticoagulant effects of their own saliva. So, wow, I've got so many questions. So first of all, how the heck does a project like that happen in the first place? Are you guys thinking about a follow-up? Are people thinking about using these anticoagulants clinically? And can you show me your Komodo dragon collection next time I visit the Gladstone?
So I don't have a Komodo dragon collection, unfortunately. I do have a few alligator and turtle embryos in methanol in the freezer. That was for a related project. I mean, how did this project come along? So it came along from an observation, again, related to understanding congenital heart disease, and again related to the dosage of TBX5. This is sort of a thread that's been following me around for 25 years now. We made an observation a long time ago. We published this in 2009, where we deleted TBX5 from just a segment of the heart where there's the junction between the left and the right ventricles, which is where the ventricular septum forms, right? And we found that when we deleted TBX5 from there, we lost entirely an interventricular septum. So we went from a 4-chambered heart, like mammals and birds, to a 3-chambered heart, which is like a frog. And so we thought, gosh, maybe there's a pattern of TBX5 in evolution that dictates the emergence of the interventricular septum. So frogs don't have a septum, crocodilians and birds do, and mammals do. So ventricular septation has evolved twice in evolution, and other reptiles like turtles, for example, which we initially examined, have a partial septum. It's like it's trying to make a ventricular septum, but it hasn't quite reached the ability to do that. And so when we published that, we showed that there was gradients of TBX5 expression, And so we thought, okay, there's going to be enhancer elements that are going to be regulating TBX5 expression. Those enhancer elements might have changed during evolution, and maybe if we can identify them, we can find what the regulatory elements that have evolved to form the four-chambered heart. Wouldn't that be cool? And so to do that, wanted to do comparative genomics. To do comparative genomics, you need genomes. And so at the time, so this is in 2009, at the time we're thinking about this, there was a gator genome that was being done, but it was poorly assembled. The green animal genome, so a lizard, turtle genomes were coming online. We participated in one of those a long time ago. And the one branch of lizards that was missing was the varanid lizards. So the varanid lizards occupy a very special place in reptilia, in that they have an almost completely septated heart, so almost but not quite, and they have a metabolism, as you pointed out, that's really, really interesting. So varanid lizards, unlike other reptiles, when they're in a hurry, when they need to run after a prey or go long distances, they can ramp up their cardiovascular metabolism to near mammalian levels. So that makes them really, really interesting. They're like quasi mammals, or they're really weird. And so we got in touch with some folks, and we could have picked an easily accessible monitor lizard that somebody's studying in a physiology lab down at UC Irvine, but we thought, go big or go home. Why not go after the biggest one out there, the Komodo dragon? It captures everyone's imagination. We also hoped that we could learn something about gigantism, because the varanid lizards also have this amazing range. They can be as big as the 200 kilo Komodo dragon, or they can be as small as a 200 gram monitor lizard. There's got a huge range of size. And so we thought maybe we could understand something, and nothing came out of the genome related to that. And so the person I started working on this, Alicia Holloway, who was our director of the genomics core at the time, she had a friend at Zoo Atlanta, Joe Mendelson, who had Komodo dragons. And so she contacted him and said, "Hey, can I get a vial of blood?" And he was like, "Sure, write up this protocol and we'll send you blood." And so, within a couple of months, we had dragon blood in the lab, and we're like, all right, let's start sequencing this. We didn't have a budget for this. We didn't have a grant for it. We didn't have people on it. So it took 8 years for this project to go along, because there was gaps of 2 years where there was just nobody, or a year or two where there's just nobody working on it, because we're like, okay, let's get it sequenced. And then we got it sequenced, and where do we get money to get it sequenced? And so, eventually we had, Poi Kwok at UCSF, who got really excited about it, and he has a genome center, and he was like, all right, we can help you with some sequencing runs, and we can help you with the mapping. And then it was like, well, who's going to do the assembly? And we're like, I don't know who, And then we happened to know somebody who had done the animal assembly, who was in the lab next door, and so he helped out. So now we had an assembly, and we're like, well, what do we do now that we have an assembly? We have to analyze this genome, and so we had nobody to analyze it. Finally, sort of pinch hitting at the, was, in the 9th inning with all bases loaded, we had Abigail Lind come in, and she's like, well, I know how to do this, and I'm interested in doing it. So she did all the downstream analysis. She discovered all these adaptations that were hidden in the genome. And that's the wonderful thing that I learned about doing this. Two wonderful things I learned about doing this project. One is the generosity of colleagues that we have around us. That famous phrase from the movie, "I've always relied on the kindness of strangers," and I won't do the Blanche Dubois voice. I'll just leave it at that. It really was a roots effort. It really was, community effort to get this done. And so that was really fantastic. And the other thing was that if you ask a genome to reveal its secrets, you're going to get not necessarily what you asked for. You're going to get what it allows you to see, and nothing more. And it was super exciting to see all these things related to cardiovascular function, and the huge signal on mitochondrial function and metabolism and generation. And it was right there. The answer was right there. It was like, bam, muscle energy. That's exactly what explains everything. It was really, really cool. The thing about the coagulation, that was really unexpected, because we kind of thought about that afterwards. We're like, yeah, these guys take down their prey by basically creating septic shock and absence of coagulation, apparent coagulation. Why is it when they're fighting with one another, over territory or over mates, why don't they fall prey to that sort of thing? So it's intriguing. It's a testable hypothesis. We're not sure that it's true, but it certainly was an interesting signal that came out.
And after all that, you can say, like not a lot of other stem cell biologists, I would bet, that you have dragon blood in your -80. So go for yours, my man. It's amazing.
Oh my God. We had dragon heart. Actually, our RNA-seq that we used was from heart. So we had an actual piece of dragon heart sitting in the freezer for a while. That was really neat.
You got to watch out for
what comes next is, we're hoping to go after those enhancers, but we realize that if we really want to go after those enhancers, we really need the epigenetic information. We really need the ChIP-seq data for the histone modifications on the tissues that we're interested in. So that would mean getting monitor lizard embryos and doing all that sort of thing. And at the moment, we're just not prepared to do that.
Not ready for all that. It's not a surprise to me, though, that you dabble in Komodos. Arun is surprised. I'm not surprised, because I have heard you quoted as saying that your passion for science began with your hands-on lab course that used axolotl. Axolotl, which famously regenerate their hearts after injury, and maybe that's what got you hooked. Is that just a coincidence that the heart regeneration in that intro to science, or is that really the root of it all for you in terms of your passion? And what's going on with that in terms, at the GLAD Center in your lab personally, tapping the regenerative potential? You just were talking about how this septal, the septum was evolved independently twice, and it's just this kind of modular, maybe gene insertion or enhancer function. Do you think that in a similar, or maybe not similar, but do you think there's a way to restore the regenerative potential of the heart in humans, or is that far-fetched?
Well, I'll answer is there a regenerative potential in the human heart? I'd say optimistically, yes. But practically, how do you do that? With gene therapy, with drugs, with all these things, it's all about delivery and efficacy. And delivering a gene therapy that would regrow a heart in a fetal human is at the moment a bit far-fetched, but it's not science fiction. It's something that I think might be a possible avenue. Now, if we go back to the axolotl in John Armstrong's third year biology class, I wasn't quite that young. I just became absolutely fascinated with developmental biology at that moment. It's just something that really grabbed me, and has always been something that I've been fascinated with. So we had an experimental lab where we used the axolotl embryos to recreate some of the famous early embryology experiments, like putting endoderm next to ectoderm and seeing an inductive power of the endoderm on the ectoderm, or taking organ primordia and transplanting them and seeing the actual organ grow in an ectopic location on the embryo. So, for example, we could take the gill primordia of the axolotl and transplant them to the flank, and then have perfectly normal-looking gills. So the notion that there's both plasticity and stability in a developmental program is something that really grabbed me right away. And so to understand the development of the heart, which is what I got interested in around that time, the notion that both the heart comes together properly 99% of the time means that there has to be some exquisite stability and precision to those gene regulatory networks. And so that's what we're at the moment trying to understand. So how does the ventricular septum form perfectly and in sync with the valves and the outflow tract 99% of the time? What are the gene regulatory networks? And we've got some really, really cool projects where we're using lineage tracing in the mouse to identify a very specific subset of progenitors that we now know give rise to only to that boundary between the left and the right sides of the interventricular septum. So we know where to look, and we're now about to understand in the next sort of few months or so, the molecules that are involved there. Now, using that knowledge, could we instruct a poorly formed interventricular septum to form properly? Maybe. It's a big maybe. I think what Arun was bringing up with the sort of the epigenetics. If we think of the mutations or the processes as being a quantitative impairment of some epigenomic pathways, I think if we can think of ways where we can tweak those, and those are highly druggable targets. It's not even just druggable, but they're very much regulated by metabolic pathways. So you can tweak metabolism and affect epigenetics in a way that then broadly affects gene regulatory pathways. And we're beginning to understand, we as a field, not in my lab, as a field are beginning to understand what are sort of the metabolic pathways, the specific ones that are involved in modulating positively or negatively various enzymes that are involved in modifying histones. So if we sort of step back and think, back to, let's say, neural tube defects and folate deficiency and folic acid supplementation in moms, that's now reduced the incidence of neural tube defects by 75%. If we could find a sort of similar thing, a vitamin-based sort of modulator of epigenetics that would mitigate or dampen the effect of the impaired gene regulatory networks that then cause congenital heart defects, I think that would be something that might be tractable in terms of modulating sort of what's happening. In terms of using cell therapy or gene therapy to induce growth, that would have to be something that would be very, very, very specifically controlled, because of course, too much of these transcriptional regulators is also not good for the proper development. I think the regenerative potential of either gene therapy or cell-based therapies is better suited to adult disease, to either post-myocardial infarction, restoration of dead tissue, or improved heart function in patients with heart failure. And we're seeing advances. And Deepak probably talked about that in his podcast, and his lab has certainly made advances towards that.
Yeah, certainly there's a lot that we need to still learn and figure out when it comes to adult human cardiac regeneration. And it's cool to see, where your inspiration comes from, because I think as stem cell biologists, we're all kind of developmental biologists at our core, right? And so we've actually had a bit of a shared road, Benoit, a bit of a shared scientific road, in that we're both, stem cell biologists using iPS cardiomyocytes to model disease, but we've also both done postdocs in the shared lab of two of the greatest cardiac biologists of all time, John and Cricket Seidman, over in the Harvard Medical School. So John and Cricket, who are a husband and wife team, have had their lab in Boston for decades and have trained a ton of scientists like ourselves. And they've been instrumental in figuring out the genetics of cardiomyopathy and cardiovascular disease. And like you mentioned, when you were a postdoc, you helped uncover how TBX5, which is a critical cardiac transcription factor, contributes to Holt-Oram syndrome. And I'm pretty sure I saw some of your reagents from the 90s still in the Seidman lab freezers. They don't really throw anything away. So that was kind of cool to see. It's like a walk through a museum in there sometimes. Yeah. So mentorship is, of course, everything in science, right? And you're actually still collaborating with them. So how have they influenced you as a scientist and helped shape you into the scientist you are today?
Oh my gosh, yeah. I've been working with the Seidmans for now, I guess, since 1996. So however many years that is, that's many years. And they've really shaped how I am as a scientist in ways that go beyond simply being a postdoc mentor. They've been cheerleaders for me. They've become friends, close colleagues. It's been really incredible that over so many years, they've moved from, I've gone from being, a naive PhD student starting a postdoc, not knowing how to clone properly, to, now where I am in my career. And every step along the way, they've been supporters of me personally and my career in some shape or form. And now we've for the last 11 years we've been close collaborators on a project called the Bench to Bassinet Project. It's funded by the NHLBI. And at first I wasn't sure how I'd feel about being on conference calls every 2 weeks with John and Cricket and sharing my most intimate data secrets. And it turns out that's been the most fun thing and the most sort of productive interactions that I've ever had. And over those decades that we've been working together, I've only gained increased respect for the two of them, both as scientists, as mentors, and just as people. They're really extraordinary individuals. And you and I, Arun, are I think really privileged to have been under their wing for at least a few years.
Mm-hmm.
That's very sweet, you guys. And I wish I were. I'm going to go do a postdoc in that lab. Hey, you guys taking postdocs? While we're on the personal note here, you've shared in the past, I don't want to impinge, but you've shared in the past that your own daughter, Nika, was born with a septal defect, making your work an incredibly personal commitment. Can you tell us a little bit about that? Knowing what you know, how do you take that diagnosis? And I've always admired your strong commitment to basic. We talked about it earlier. You guys go deep on cardiogenesis. Do you think that your daughter's experience has driven you to place a greater emphasis on the clinical translational application? I know you started in the outset saying that the whole end game is translation. It's basic mechanism, but toward an applied clinical goal. Did that shift? Are you more motivated? I know in your case, it's not an issue anymore, but knowing or having that experience, you can really empathize with a lot of parents out there that are going through a similar experience. Does that motivate you in part?
It really does. I mean, up until then, I'd written the chapter or the reviews on the etiology of congenital heart disease, and after the echocardiography, where I actually saw the Doppler jet going across the interventricular septum of my daughter, who was squirming and not letting the echocardiographer. She was 2 weeks old. It was at her two-week checkup that the pediatrician was just listening in, and I was looking at him, and I was like, he's with a stethoscope, and I'm like, he's listening way too long. What's going on? And anyway, my wife asked me, she said, well, what did we do wrong, and what causes this? Why is this happening? And I didn't have an answer for her. I said, I don't know. And I said, and I thought, gosh, despite, I've been studying this for, at the time, I'd been studying this for 15 years. I'm like, I don't know. I actually don't have an answer for you. And so in a sense, it really did motivate me a great deal. I mean, scientifically, I was already very motivated, but it really kickstarted my motivation. And it also made me realize, that when we talk about 1%, right, 1% of live births have a congenital heart defect, you sort of think, well, that's not a lot. That's not a lot. But when you're in that 1%, and I like to joke, we're in the wrong 1%ers. I'd love to be in that other 1% group. But that 1%, that counts for a lot. And when you think of chances of, let's say, of developing a particular type of cancer, where it's like, it's 2% if you have such a mutation, that 2% counts for a lot. And so, and definitely, and as you alluded to it, the VSD closed by itself, and so we got out of the woods, after a year and a half of a bit of anguish. But there's a lot of parents who don't. A lot of parents who have much more serious heart defects, and in fact, we're working with a foundation that's been established by a family in Silicon Valley whose child has a very, very serious heart defect, who's had 3 open heart surgeries, and they desperately want to understand how this happens and what you can do about it. Because the other thing about congenital heart disease that a lot of people don't think about, but despite the success in surgical correction, there's a lot of things that come along with the structural defect, right? These kids have degrees of arrhythmias. They have they develop heart failure. They develop diastolic dysfunction. They have other sequelae that we don't understand, that we don't necessarily know if it's the mutations that directly caused that. Those have in the past been ascribed to, oh, well, this kid had complex surgeries, has had ischemia, has had a number of things, so it's probably secondary to that. But we think there's a lot more, and we know that genes like TBX5 that we talked about are directly involved in aspects of heart function that go beyond the formation of the heart. It's a major genetic risk factor for atrial fibrillation, for example, and is directly involved in regulating the contractile properties of the heart. And so even though you might surgically correct the heart, there are other things that we need to understand and that we need to treat better in these kids with congenital heart defects. So all that to say that, that this personal link has definitely touched me in many ways. I mean, the whole initiation of me wanting to study the heart was my father's myocardial infarction when I was 16 years old and he was 43. He survived it, had triple bypass surgery, and he's now in his late 70s and has an ejection fraction that is well below, so his heart function is severely impaired. He has a pacemaker defibrillator that once in a while keeps him alive, and there's nothing we can do about him. And his failing heart, and so that's another thing that motivates me tremendously, and so I've got these two people in my life that I love dearly who've had two very different spectrums of heart disease, and I think of them with every experiment that we do in the lab, and despite the focus on basic science. I hope at some point I can do, something useful for my dad and something meaningful for my daughter, and for everybody else out there who has these, issues that seem unlinked, but actually are just two ends of the same spectrum.
Yeah, thank you for sharing that, Benoit. And heart disease is something that unfortunately affects so many of us and so many of our families. It's also my own motivation as to why I got into this field. So everything that we do definitely helps. And we're approaching the end of our interview here. And so before we let you go, we're going to ask you a few non-science related questions. One sort of science related question. It's no secret that you're really active on Twitter. Okay. And a lot of other academic folks, not including Daylon, have become pretty active on Twitter over the last few years and using it for science communication and a little self-promotion here and there along the way too, right? But in addition to the usual science communication, I've noticed that you're actually very vocal on Twitter about important non-science topics like social justice, diversity, and even politics. So talk about your approach to using Twitter, how you got into it, and how you use it not only for science, but also to talk about these other important issues too.
Yeah, I got into it. I'm not sure how I got into it. I guess it's just something that, I got interested in and saying, hey, what are people talking about on this platform? You sort of hear about it. And this was quite a while ago. This is about 10 years ago or 9 years ago I started on it. And I got really excited in the scientific community, especially the folks in genetics and genomics were early adopters of Twitter and were a very interactive group. And I learned a ton of both technical things and there was a whole thing about people live tweeting conferences. I was like, wow, I can follow somebody's Twitter thread about this genomics conference that I'm not attending and get a ton of information in real time. The other, I found myself at one point, I was having a scientific discussion with Mike Eisen, who was over in Berkeley, which is he was a couple of miles away from me, somebody in the UK and somebody in Japan. And in real time, we're having this nitty-gritty discussion about some science topic. I thought, wow, this is a place where I can interact either in real time or almost in real time with people around the world who I've never met and who have really interesting views on things and initially on science. The other thing I really like about Twitter and partly why I engage with it the way that I do is that it is a great equalizer. I just had a I just had a conversation about somebody's paper just yesterday with, the senior author on the paper, with, another, scientist over in the UK, and with a graduate student in Ottawa, Canada, in the same department where I did my undergrad. And, and all of us are discussing this, like, like, it doesn't matter who we are. It doesn't matter that I'm a director of a research institute. It doesn't matter that the senior PI is the PI on the paper is a junior PI. It doesn't matter that it is a graduate student in a small university. We're just talking science. So it's an amazing equalizer, I find, that there's no hierarchy, or at least there shouldn't be. And those who display some kind of notion that there's a hierarchy really don't do very well on Twitter, because everybody is on par, or at least should be. And about other issues, I view Twitter as something that should reflect my voice and my personality. Sometimes that's a bad thing, and sometimes that doesn't come out quite right, just like it does in my normal conversations. And when I have strong opinions about social justice, I share them with my friends, and I don't know who's necessarily who's listening on Twitter. That's one thing that amazes me, is that people actually pay attention to people like me. But I don't know, I like having conversations about things that are important to me, just like we're doing with this this podcast. I really like having these conversations about things that I think are interesting, or things that are important. I try to stay away from politics, because I realize that's an overly touchy subject. Sometimes it's really hard not to, especially these days. Social justice, though, that's something that's always been really important, especially now.
Yes, especially now. And yes, you're facile with the new media, maybe more than some people who are on this podcast right now. I'm looking to get on Twitter any day now. I'm thinking about it. I'm wrestling with it. But let's move away from the new media to the old media. If we're talking science peripheral question, let's talk about, the old ink and paper. What's a non-science or science-related book that you're reading, you've read, that is awesome and that you would recommend to our listening audience?
Yeah, one of my favorite books in the last, I think, 10 years is Viet Nguyen's The Sympathizer. It's I don't know if you've read this. Viet is a professor. I can't remember where he teaches. He's an American. He's a Vietnamese immigrant, and he wrote this book about Vietnamese immigrants right after the Vietnam War, and it's written in a voice and in a style that's really absolutely captivating. The story is super interesting. It's a story about a Vietnamese informant who then escapes Vietnam and establishes himself in the United States, and his family, and how they want to and organize a return to take back over Vietnam, and it morphs in the end where the individual goes back to Vietnam and is captured and jailed for having been an informant, and all of a sudden the book, and he's forced to tell his story and the book changes from an anonymous narrator to now this person telling his story, and it's just a literary masterpiece and a fantastic view into the immigrant experience in the United States in the early 1970s. It's really, really wonderful book.
It really resonates right now. That's Viet Nguyen, who is a professor at USC. Just look that up for you. Yep. And finally, to close, will you just tell us who, you're a hero to a lot of young scientists out there. Who are your own personal scientific heroes?
Yeah, so I've got two. I mean, we mentioned the Seidmans, and they've been fantastic mentors to me. I'd say probably my biggest scientific hero is Janet Rossent, a very famous developmental biologist who spent most of her career in Toronto. She is a force of nature, And she has always been a very strong woman in science. I think she should be the exemplar to women in science. She was a faculty at a small university in Ontario at Brock University at a time where women scientists were not, celebrated or not doing as well as their male colleagues. And she built a career on early mouse embryology at a time where very few people were doing that. And she had the wherewithal to sort of propel that towards, again, using basic science and embryology to understand principles of disease, to understand molecular mechanisms, and really was one of the leading lights in Canada and the world in the use of mouse embryonic stem cells, first in making mouse knockouts, but then in using them to understand fundamental properties of pluripotency and lineage allocation. But beyond the science, she's been an extraordinary mentor to a number of people, not just in her field, but across almost all disciplines. She was the director of research at the Hospital for Sick Children when I was there, and continued growing that institution into something that was absolutely phenomenal. She's been a leader internationally, for example, in the ISSCR and many other organizations. She's been an enormous influence for women in science and for just about anyone, young person who wants to succeed in science. She mentored me and helped me succeed. And she's, somebody who's touched so many people while at the same time doing the most incredible biology that has influenced hundreds of people in many, many disciplines.
Yeah, and I will throw in there that she's top 3 interviews we've ever done on this show. I mean, might be number 1. My favorite chat that I ever had. Like you said, a force of nature. Not unlike yourself, although maybe you got a little bit more growing to do. Dr. Bruno, it's been really nice talking to you. Thank you so much for joining us, and we'll be sure to have you on again if you're willing.
Thank you for having me. This has been really fun.
All right, you guys, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on Twitter at StemCellPodcast or by email at info@stemcellpodcast.com, where you can give us some feedback or suggest guests. Like I said in the intro, we have 3 great interviews with some of the top stars of the ISSCR coming up in the next few episodes, so listen close to that. Thanks, you guys, for joining us for this one. It was a great episode. Thank you, Benoit, for your wisdom. We'll see you guys next time.